So we are interested in aneuploidy in cancer and copy number change, which we've heard about today. And this can happen as a whole chromosome amplification or deletion, or smaller focal amplifications or deletions. And in some cases, we think we know what genes are important in those recurrently mutated regions, and sometimes we don't. And I'm a hematologic oncologist, and trisomy 21 has fascinated the hematology community and all of medicine for quite a while. And so we studied B-cell ALL in the lab, and you may know that in Down syndrome children, there's a 20-fold increased risk of B-cell ALL. But in non-Down syndrome ALL, among kids and adults, gain of chromosome 21 is the most common aneuploidy that occurs. There's also a phenomenon called IAMP21, which is a small focal amplification of the distal arm of 21, which is recurrent in BALL. Is a very poor prognosis. There were just 2 papers in JCO last week about this. It's basically the poorest prognosis cytogenetic change in pediatric ALL. And then in hypodiploid or near haploid ALL, so these are leukemias that lose chromosomes, they never lose chromosome 21. So there seems to be a dosage balance about this chromosome 21. And then it's not just simply a random occurrence. It seems to occur in particular ALL subtypes, these that have the CRLF2 or JAK2 mutations, kinase active, or the so-called BCR-ABL-like, so they have a gene expression profile like BCR-ABL, but they don't have the BCR-ABL translocation. And we did an analysis at the Farber, and plus 21 was present in a significant proportion of relapsed ALL. I just want to highlight this recent paper about the hypodiploid ALL, and we were very struck by the fact that these are tumors that have basically 24 chromosomes. They've lost every copy of one of the chromosomes except 21 is just never lost. So there's really something about a balance between something on 21 and ALL. So we're hypothesizing that overexpression of one or more loci on chromosome 21 by this amplification event may contribute to lymphoid leukemia pathogenesis, and one may cooperate with a kinase active signaling and may create targetable dependencies. So the biggest sets of Down syndrome-related ALL have not really come up with a unique signature of what defines a Down syndrome or a plus 21 leukemia. In fact, this group said that it's a highly heterogeneous disease and they couldn't define it as a unique entity amongst other ALLs. So what we did instead was to start with mouse models. And this is a schematic of human chromosome 21, and here's that IAMP21 segment that is a recurrent abnormality in human ALL. And it turns out that many groups have made mouse models to try to study Down syndrome. And this is the smallest triplicated mouse. So this is an engineered mouse to have 3 copies instead of 2 of these 31 genes that are on mouse chromosome 16 that correlate with human chromosome 21. So this is called the TS1-RHR mouse, and it triplicates the so-called Down syndrome critical region, because in kids with partial trisomy, it seems that this is the minimum amount needed to have Down syndrome phenotypes. So I'm going to summarize mouse models here on this mouse model on one slide. This was a lot of work, but basically we could use this mouse to show a few things. When we took B cells out of this mouse from the bone marrow and put them into colony assays, they basically adopted a transformed phenotype over passages. So the trisomy B cells would just start to grow out indefinitely, essentially, whereas the wild type cells died out after a few passages. There was also a differentiation abnormality in the B cell differentiation in the bone marrow of these mice. This paralleled a finding that was recently described for human Down syndrome fetal liver hematopoiesis, the same stage of B cell differentiation as a partial block. And then we could combine this in the mouse with either BCR-ABL on the top, or with a very complex 5-way mouse model we made to model the BCR-ABL-like leukemia that has these several genetic abnormalities. And in every case, when we add the trisomy 21 model to the other model, we get faster leukemia and more leukemia. So we think now we have a model that replicates some of human disease, but narrowed down to only 31 genes triplicated. So this is just 3 copies of 31 genes instead of 2, leads to all these phenotypes. And now we can ask whether we can use the model to define a signature that we may be able to go back to patients with and learn something about the disease. So we did RNA sequencing, and this is network enrichment mapping of a gene set enrichment analysis from comparison of trisomy B cells to wild type B cells from the mouse, mapped back to human data sets. And we found some interesting things, namely that stem cell, cancer, lymphoid differentiation phenotypes are the kind of gene sets that come up when you compare the trisomy to the wild type cells. But also there was this node of polycomb repressor complex 2, or genes marked by H3K27 trimethylation. So we've heard all about that and what that means. That's a repressive mark, and we'll talk about that in a second. But that was striking to us. So as we've heard, the PRC2 complex is a repressive complex, if you will. It adds this methyl mark to H3K27. Genes that have this mark are repressed. But this is quite complex. So we heard just now about gain-of-function mutations in large B-cell lymphoma and follicular lymphoma. But as Tim Grobert mentioned, there seem to be clearly loss-of-function mutations in myeloid diseases and in T-cell ALL. So it's sort of unclear the balance of what's happening with this pathway in hematologic malignancies. So we looked back in our mouse, and we collaborated with Jake Jaffe over at the Broad Institute, and we did histone mass spec, and we found that indeed on a global level there is decreased histone H3K27 trimethylation in the trisomy B cells. We did a lot of chip sequencing to show that actually on a global level, especially at transcriptional start sites and in gene bodies, there's a decrease in H3K27 trimethylation. And in the leukemias we generated in the mouse, there's a very striking global decrease in H3K27 trimethylation. So it seems to be somewhat characteristic of leukemias that have an extra copy of these 31 genes. And when we took signatures, multiple signatures, either signatures generated from our mouse B cells or some of these polycomb repressor complex target signatures, and went back to patients with ALL and did unsupervised clustering, these signatures can distinguish the Down syndrome leukemia patients from the majority of non-Down syndrome leukemia patients. And we can even take this to patients without Down syndrome that have just somatic gain of polysomy 21 or that IAMP21. And in this relatively small data set, it was still able to cluster those patients together using these same gene signatures. We want to talk about therapeutics here today. So we tried to use both genetic and chemical experiments to determine whether B-cell growth of these B-cell progenitors is actually controlled by this H3K27 methylation. So we took advantage of a couple of Glaxo drugs that we could buy that modulate H3K27 methylation in two directions. One is an EZH2 inhibitor that we heard about. So when cells are treated with this inhibitor, you decrease the methylation, right? Or if you inhibit the demethylases, you would increase the methylation of the cells. So remember, our cells have a defect where they seem to be hypomethylated. So our hypothesis was that if we could restore methylation by sort of tilting the balance in the opposite direction, we might have an effect on growth of these cells. So indeed, that's what we found. So we took our cells straight out of the mouse. They're not transformed yet. We don't see a huge difference in the trisomy cells. But when we grow the trisomy cells in culture, remember in red, they grow like crazy, but we can block that by treating with the demethylase inhibitor. And sort of as a proof of principle, we did the opposite experiment just for fun, and this kind of gets to what Tim was alluding to before. If we just take wild-type bone marrow cells and put them in our colony assay, they die after 2 passages. But when we treat them with the EZH2 inhibitor, they grow forever. We can grow them for several months at this pro-B cell stage in the presence of an EZH2 inhibitor. I'm not going to talk more about that today, but I think that has some interesting implications for using these drugs, but also if there are potentially therapeutic uses for expanding B cells. Now we want to treat patients. So these H3K27 demethylase inhibitors are not great drugs. None of them can be given in vivo, even in a mouse yet, because they're eaten up by plasma esterases, and the ester group is necessary to get into cells. But in vitro, this is a whole collection of human ALL cell lines, and they're all sensitive at less than micromolar sensitivity. Some are more sensitive than others. There are unfortunately no Down syndrome cell lines, so we can't really prove our hypothesis there. For what it's worth, we threw in a diffuse large B-cell lymphoma, Carpus 422, that has an EZH2 activating mutation, sort of as a negative control. And at least for what it's worth, that was the least sensitive. It seemed shifted to the right in comparison. And we did the only experiment we really could do on primary xenograft cells, because we can't give this drug in vivo, in that we treated some DS and non-DS ALLs in vitro. And I didn't separate these out by color because I'm not trying to say that the DS ALLs are more sensitive than the non. That would have been nice, but it looks like they're all sensitive at a similar dose. And if we took one of the non-DS ALLs at a dose where there is no difference in cell number after 3 days of treatment and then transplanted into recipient immunodeficient mice, we do see, in fact, decreased numbers of cells in the bone marrow and spleen, suggesting that this may have some therapeutic efficacy, but we need basically a better drug before we can prove this further. And I'm going to just give you a little interesting hint that may not have been in the abstract about what genes we think are important for this, because now we've narrowed it down to 31, we can ask this question. So right off the bat, when we knew about this epigenetic phenotype, we looked at the genes. Here's the 31 genes, and here's their expression across 1, 3, and 6 passages in vitro. So we were looking for a gene that maintained its expression through all the passages, and there were several of those. But this one, HMGN1, kind of caught our eye. So HMGN1 binds to nucleosomes. It does the opposite of what histone H1 does. So histone H1 helps compact nucleosomes. HMGN1, when added to a milieu, opens up nucleosomes. And when we looked back at our patients, the patients that clustered in this cluster with the most of the Down syndrome patients had the highest HMGN1. The furthest away had the lowest. And in fact, the few outlier patients that did not have Down syndrome that clustered with these patients had the highest levels of HMGN1. Now this is, again, completely correlative only. But when you look at what's known about HMGN1, it tends to land on ChIP-seq at regulatory regions of genes and is sort of mutually exclusive with H3K27 trimethylation. So it goes where genes are active, and it excludes H3K27 trimethyl. So we could take pro-B cells and overexpress HMGN1 dramatically so, not just 1.5-fold like in our trisomy, and we can actually drive down the K27 trimethylation, and represented here by this plot. So it seems that when we overexpress HMGN1, we may open up chromatin, resulting in lower H3K27 trimethylation. And when we did an shRNA interrogation of those 31 genes with 5 hairpins for each gene, the far and away hit for genes that were selectively toxic more to the trisomy cells than wild-type cells when knocked down was HMGN1. So we got our hands on an overexpression mouse for HMGN1. This is our last slide. And basically, when we take B cells out from that mouse that overexpresses HMGN1, they have an increased growth in vitro. It's not sufficient to grow forever. So something else among those 31 genes is important. But nonetheless, these cells recapitulate the signature of the full trisomy cells, including the PRC2 target gene signature. And when we combine that mouse with BCR-ABL in a leukemia model in vivo, we again have a dramatic acceleration of the BCR-ABL leukemias. So HMGN1 is contributing to this phenotype, but may not be the only important gene. And I doubt that it would be, because we don't see single amplifications, we see this region. So I've shown that triplication of these 31 genes is sufficient to confer these transformed phenotypes, and that BALL with plus 21 is associated with overexpression of PRC2 target genes, so loss of the repressive mark. So plus 21 may confer targetable dependencies, as I showed, and the gene HMGN1 we think is important. So this is sort of a hypothesis slide. So I think clearly in gain-of-function mutations in EZH2, PRC2, like diffuse large B-cell follicular lymphoma, we would use an EZH2 inhibitor. But what do we use in those loss-of-function cases, where there are clearly mutations in EZH2, SUZ12, even ASXL1, which is an associated gene, all seem to result in this loss-of-function mutation. And maybe I could add to this BALL with plus 21 and maybe those other subsets. So we would suggest that maybe we can push this reaction back in the opposite direction, if you will, by inhibiting the demethylases. Or perhaps if there was a way to target this HMGN1 interaction with chromatin, you might also reverse some of the phenotypes. So with that, I'll thank lots of people that worked on this, and thanks, I didn't mention, to Bruce for having me here today.
Okay, thank you, Andrew. Any questions?
Thank you. So the lines that you talked about that were grown in the GSK126, the B cells that grew for like 3 months or so?
Yeah.
At the end of that period, were they still dependent on the inhibitor for that growth?
They were dependent, at least as far as we took them out. I'm going to show you data taking them out to about 6 weeks or so. If we remove the drug, the wild-type cells in black died off over a few passages. Interestingly, the trisomy cells, when you take away the inhibitor, they still grew. And that corresponded to how much the sort of reset baseline of H3K27 trimethylation. So we didn't completely transform the wild-type cells. I'm not sure what would happen if we went longer and longer and longer. But we've actually repeated this with several EZH2 inhibitors, including one that Jay Bradner made in the lab next door to ours. And it's a real phenomenon that I think it has something to do with this culture condition where we're driving these colonies with IL-7, and if we block EZH2 at the same time, you just expand some self-renewal capacity.
First, Andy, what is HMGN? Is it high molecular weight group protein?
N1, yes,
N1.
that is right. So there are many.
So I told you about HMG1 when you were a clinical fellow. I think it belongs to a group of proteins which identify areas of DNA distortion. Is that right?
Yeah, so there's many. There's the A family, the B, and the N family, and they're all slightly different. Some bind directly to DNA, some bind only to nucleosomes. But yes, basically they're dynamic. And so anywhere there's dynamic changes in chromatin structure, these things go there.
So apparently they're associated with platinum sensitivity as well, cisplatin, platinum complexes, drugs. That's all I know about it. You've taught me a lot.
We're going to try to figure out more about it.
Okay, thank you very much, Andrew.